Hi everyone, and welcome to my talk entitled Sex Matters, Male versus Female Drug-Induced Arrhythmogenicity. My name is Matthias Sperling, and I did this work as part of my postdoctoral research at Stanford University, and this work was done in collaboration with Francisco Sari Costabal, professor at Pontificia Universidad Católica de Chile, and Ellen Kuhl, professor at Stanford University. So to give you some background into why we looked into sex differences in drug-induced arrhythmogenicity, I wanted to take you back to this paper by Bazet et al. From over a century ago, where he analyzed the time relations of electrocardiograms between males and females and noticed important differences there. So he even concluded that the difference seen between men and women in the electrical changes must be genuine and are difficult to explain. Now, over a century later, in 2020, Carbon et al. Quantified these differences in more detail. And what we can see from their results is that almost all features in the electrocardiogram between men and women have important and significant differences. So this is already a first proof that there are important sex differences between male and female cardiac electrophysiology. These sex differences in cardiac electrophysiology also become more clear when we look at the approximate distribution or sex distribution of patients suffering from certain cardiac electrophysiological disorder. For example, what you can see here is that men suffer from Brugada syndrome way more often than women do, while women are more prone to develop long QT syndrome than men are. So with respect to that, we know that there are important sex differences in cardiac electrophysiology. Nevertheless, for a very long time, science, and I will refer to this as old science in this talk, referred to women as being similar to men just without the hormones. Now, of course, we know that this is wrong to this day. However, due to this way of thinking, or at least this old way of thinking, the female sex is highly underrepresented in animal trials, and women are highly underrepresented in clinical trials. And due to this lower representation of the female sex or women in these trials, there is an important sex bias that has developed over the years and is still present to this day. And this sex bias can actually have dangerous consequences. So if you think about these sex biases and the consequences it can have for the safety of drug usage, for example, one of the probably most notorious examples of this is the Zolpidem story. So Zolpidem is a sleep drug that has been used for multiple decades already. But for over 20 years, the dosage used of Zolpidem was the same for men and women. However, back in 2013, novel evidence arose that proved that women actually metabolize this drug slower than men. And due to this, and due to the fact that most of the tests were done on men, the women were typically overdosed. So women often suffered from next morning drowsiness, substantial cognitive impairment, and increased traffic accidents just because they metabolized the drug slower and they still had quite some active drug in their system. And in 2013, after this evidence arose, the FDA finally halved the recommended dosage prescribed to women compared to men. However, when this story arose, a lot of people actually already realized this was probably just the tip of the iceberg. Indeed, if we look at multiple drugs and their risk for acquired long QT syndrome, we can actually see that many drugs have a greater risk of inducing arrhythmia in women than in men. However, the research on this topic is rather limited, and we don't really understand why this is the case. So in general, we can say that the differing risk for drug-induced arrhythmogenesis between sexes is dangerously understudied, and its mechanistic origin remains unclear. This problem and to provide a mechanistic insight into the sex differences in drug-induced arrhythmogenesis, we used a multiscale computational framework in which we can integrate sex differences at the ion channel, cell scale, tissue level, and organ scale, and then look at what the differences are and how these sex differences propagate across the scales to potentially cause arrhythmias at different dosages for different drugs. So in more detail, we developed a multiscale cardiac exposure response simulator that allows us to integrate processes at multiple scales to look at arrhythmias. So at the subcellular scale, this requires us to solve ordinary differential equations of the temporal evolution of voltage-gated ion channels and how that leads to the action potential developing in the cells, so the depolarization and the repolarization. At the tissue scales, we solved the spatiotemporal evolution transmembrane potential convection diffusion problem to see how the signals travel throughout the tissue. And in that, on that scale, we also take into account the anisotropic organization of the tissue so that the signal travels at a different speed along the fiber direction and across the fiber direction. And then at the organ scale, we bring all these processes together and include the tissue heterogeneity and anisotropy in a subject-specific 3D geometry of the human heart. So to then develop sex-specific multiscale cardiac exposure response simulators, we dove into the literature and combined all the data that we could find on sex differences at the multiple scales. So for the subcellular scale, we used experimental quantification of sex differences in cardiac ion channel activity of non-diseased human hearts, where we used the mRNA expression of genes encoding ion channel and transporting subunits and the protein expression of these subunits themselves as well. We used those to scale the conductivity of different ion channels in the male and female cardiomyocytes. Doing this, we developed male and female cardiomyocytes models in which we can look at the action potential evolution over time. You can see the result of that here, and you can it's pretty clear already from these plots that there are important sex differences at the subcellular level, where the male cardiomyocytes typically depolarize and repolarize faster than the female cells do. And in the endocardial cell, you can see the comparison of these two male and female cardiomyocyte models compared to the baseline O'Hara-Rudy model, which was based on 54% male and 46% female data. Now, at the tissue level, we incorporated tissue conductivity differences by looking at the experimental quantification of the connexin 43 protein, which is the principal ventricular gap junction protein that is responsible for conductivity in the tissue. And the results of that can be seen here. So there are also important conductivity differences that can be found in the cardiac tissue between men and women. And then at the organ level, we took into account the size differences between male and female hearts that were reported in literature to actually develop a male and female 3D heart model, as you can see here on the right. So bringing this all together, we have two cardiac electrophysiology models that combine information from the ion channel scale all the way up to the organ scale. And the result of that can be seen here. So you can see that the differences just in the healthy case are pretty small. However, it takes a little bit longer for the female heart to repolarize, which is something we did expect. And then looking at the cardiac electrocardiogram that we can compute from this, we can see that the most important difference we see here is in the QT interval. So the T wave happens a little bit later than it does for the female heart than it does for the male heart, which is also consistent with what we have seen in the literature. So now that we have these sex-specific cardiac exposure response simulators, we can start looking at the sensitivity that male and female hearts have to drugs. So we know that drugs can bind to each ion channel and modulate the channel behavior, which we can incorporate by bringing these fractional blocks into our models and by quantifying the effect that a drug and specific drug dosage has on blocking the ion channel in general. However, if we want to do this for each possible ion channel block in full multiscale models, we actually have a sensitivity study that's not really computationally tractable. And therefore, we focused on the 7 most important ion channels for this study that were already identified by the CiPA initiative, which you can see here. So what we did is that we took our male and female cardiomyocyte models and we performed Latin hypercube sampling of a 7-dimensional parameter space of potential ion channel blocking combinations, which you can see here. And then we simulated the sex-specific mid-wall cell behavior for each of these combinations. And then we looked into the fact whether or not there are early after depolarizations or not, because that is a sort of surrogate for the risk of developing Bursata de Pantes or arrhythmogenesis in general. So as you can see here, for this 10,000 cell simulations, we already have the first important note to make, is that in the male mid-wall cells, we see way less early after depolarizations than we see in the female mid-wall cell. Now, we can look at each of these simulations and then quantify the presence or absence of early after depolarizations and use logistic regression to actually quantify which ion channels, or which drug-induced ion channel blockings at least, are most important in causing these early after depolarizations. So we did that for both the male and the female mid-wall cell, and we could conclude from this that drug-induced rapid late rectifier current blocking is the most pro-arrhythmic for male mid-wall cells, while that's also the same for the female mid-wall cells. Whilst on the other hand, drug-induced blocking of the L-type calcium channel is the most antiarrhythmic for the male mid-wall cells, which is the same for the female mid-wall cells. However, this is not a non-result, because there are relative differences in how much these drug-induced ion channel blockings have an effect on causing early after depolarizations. So now that we quantified the two most important drug-induced ion channel blockings for the development or the non-development of early after depolarizations, we wanted to do this on our full multiscale models. So what we did here is develop a two-dimensional parameter space that looks into the drug-induced ion channel blocking of the L-type calcium channel and the rapid delayed rectifier current. And what we then have is a parameter space in which we want to quantify the arrhythmia risk. So the normal way of then filling in this risk space is then to do random samples and then looking at the risk in the multi-scale model, whether or not we develop torsades de pointes or arrhythmias. So you can see here, for example, for this combination of beta CAL and beta KR, we do not see any torsades de pointes or arrhythmias in this full multiscale model, so we consider that one as safe. Then for this point, for example, we do see the development of torsades de pointes, so we can say that this combination is unsafe. The same for this point, where we can see that actually, even though we have a pretty high beta KR, there's enough blocking of the L-type calcium channel, which is antiarrhythmic, to still have a safe combination of these two ion channel blockings, while for this case here on top, that's no longer the case anymore. So we could do this for the whole parameter space. However, that's computationally very expensive, because each of these simulations is expensive on its own and requires multiple CPUs for multiple hours on a high performance cluster. So we had to be smart about the way that we did this. So therefore, instead of just randomly sampling the whole parameter space, we actually used some machine learning techniques, and I will just name them here for the sake of time, but they are explained in way more detail in our paper. So we used multi-fidelity Gaussian process classification and active learning to actually explore this parameter space and quantify the boundary where the risk boundary between safe and unsafe drug dosage combinations. So we did that for both the male and female heart. So we first did a low fidelity risk classification, exploration, and active learning exploitation of the cell scale simulations, and then we use that information to learn the multi-fidelity risk classification boundary, as you can see here, for the full multi-scale male heart models. Subsequently, we did the same thing for the female heart model. So first we looked at the female low fidelity cell simulations and quantified the risk classification boundary there. And then we used that information to learn the multi-scale risk classification boundary more quickly, and that we can easily identify the most interesting cases to simulate, as you can see here. And then we have cases where we do develop torsades de pointes, as you can see here on the left, and then the cases where we don't develop torsades de pointes, but just have a different stage of where the T wave is, for example, here on the right. So now comparing both multi-scale risk classification boundaries, we see a very important difference, and that is sort of explaining what we were after from the start and what we wanted to look into in more detail. And this difference was pretty substantial, as you can see here. So now we have a male multi-scale drug risk classifier and then a female multi-scale drug risk classifier. Once trained, we can use these drug risk classifiers to actually look into the dose-specific drug risks that we have for male and female hearts. And we started off by looking into the high-risk drug dofetilide, which is a drug that is typically used for the maintenance of sinus rhythm and for treating atrial fibrillation, but it's also known for its risk in causing ventricular fibrillation. And this drug is marketed in the US under the name dicazin. So what we can do now, or how we use these drug risk classifiers, is that we first look into the beta KR, beta CAL combination that different dosages cause, and that we plotted here by using this color bar, as you can see here. So if we go up with higher dosages, we also travel within this two-dimensional beta KR, beta CL parameter space. So now for dofetilide, we can then look for the male and female heart separately when this dose drug-induced ion channel blocking combination actually reaches the drug risk classification boundary. And then for the male heart, we saw that this is at 26 times the free plasmatic concentration, while for the female heart, this actually already occurs at 3.5 times the free plasmatic concentration. So this shows that women are more at risk of developing torsades de pointes or arrhythmias with these concentrations, as you can see here. And looking at clinical practice or just dofetilide in general, it's already important to know, and this refers a little bit back to the start of our presentation, that in original clinical trials testing the safety of dofetilide, women only account for 28% and 16% of the total amount of the 538 enrolled patients. So that's important as a little side note. But then looking at these different concentrations that we see here, if this would be correct, we would actually see that women are suffering from drug-induced arrhythmogenesis, or at least drug-induced elongation of the QT interval way more often than men. And actually, if we then look at this study, which is a 2018 clinical center study, we see that the dofetilide dosage reductions due to the onset of long QT syndrome in women and men happens 3 times more often in women than it does in men. So this sort of confirms that women are at more risk for dofetilide drug-induced arrhythmogenesis, and how this refers to or relates to our mechanistic results that we got before. Until today, still the same dofetilide dosing algorithm is used for men and women. So that's something to keep in mind. Another drug that we then looked at is the medium risk drug chlorpromazine, which is actually used for psychotic disorders. And then looking at this dosage drug-induced ion channel blocking line, as you can see here, which is color-coded, we see that for male hearts, we do not expect at any dosage that the line crosses the male risk classification boundary, as you can see here. So we would not really expect any chlorpromazine-related drug-induced arrhythmogenesis in men. However, for female hearts, we saw that this actually does happen, so it does cross the risk classification boundary at 80 times the free plasmatic concentration. So that concentration is pretty high. However, it still means that there is a certain risk. And then if we again look at the clinical literature, we saw that in 4 decades of clinical case report data looking at chlorpromazine, there's only 7 cases that were published where chlorpromazine actually led to induce some sort of arrhythmia. And interestingly, all these cases involved women. None of these cases involved any men whatsoever. And then a final case where we looked at was diltiazem, normally considered a safe drug, which is used to treat high blood pressure. And indeed, as we can see here, the dosage-dependent drug-induced ion channel blocking combinations for beta-CAL and beta-KR for both men and women never really cross the risk classification boundary. So we do not expect to see any drug-induced arrhythmogenesis from diltiazem here. So in conclusion, in this study, we studied the sex differences in drug-induced arrhythmogenesis because it's highly understudied and the mechanistic origin remains unclear. And therefore, we combined experimental cell, tissue, and organ scale data to develop mechanistic sex-specific cardiac exposure response simulators. We used machine learning techniques to explore the male and female sensitivity to drug-induced ion channel blocking, and we trained a male and female drug-induced arrhythmogenic risk classifier. Next, we showed significant sex differences in clinical drug concentrations for a high and intermediate risk drug. So with that, I'd like to thank you for your attention. And if you have any more questions, please feel free to contact me. If you want any more details on the study, you can also read up on it in the paper that was recently published in Frontiers in Physiology called Sex Differences in Drug-Induced Arrhythmogenesis. We also coupled the drug risk to the performance of the human heart and into the mechanical consequences of drug-induced arrhythmogenesis in our latest preprint on bioRxiv. So I also like to refer you to that paper if it's of interest to you, and it's called How Drugs Modulate the Performance of the Human Heart. So with that, thank you for your attention, and I look forward to discussing these results with you in more detail.